So welcome to another video by Joe Academy. Here we are again with another video on amino acids to continue from where we ended. I want to thank everyone that's been there with us, everyone, our subscribers, our viewers. We thank you very much. Please, if you're watching this video and you have not subscribed, please make sure you click that subscribe button and also make sure to smash that notification button so that whenever we upload videos you'll be notified and you will be the first to watch. Thank you very much. So let's continue with the acid-base properties of amino acids. So to start, amino acids are amphoteric. That means they have acidic and basic properties. They can act either as an acid or as a base. What do we mean by that? An acid is a proton donor, while a base is a proton acceptor. So amino acids, they can act as a proton donor or as a proton acceptor, depending on the conditions in which they are. So if you place an amino acid in low pH, that is in acidic medium, what will happen? In low pH, amino acid, it means that there is more of hydrogen ions in solution. So the amino acids now will act as a base by accepting protons. Since low pH means there are more protons in solution, so the amino acid acts like a base and accepts the protons. But in high pH, that's in basic medium, means there are more hydroxide ions in solution, so the amino acids now will donate protons by acting as an acid. So to kick off, for example, here we have an amino acid with a low pH. An amino acid at low pH here. So you can see the amino acid at low pH has accepted a proton at the level of the amine group here, and it is positively charged. So this is to tell you that amino acids at low pH are positively charged, while amino acids at high pH are negatively charged. So at low pH, for it to dissociate, when you continue increasing the pH, that's in this direction, to a high pH, that means you are adding more of hydroxide ions in solution, increasing the pH up. The amino acids begin to lose their protons as you continue adding. So there is a concept for an amino acid to lose a proton, that is dissociate. In other words, for it to lose a proton, the pH of the medium must be greater than the pKa of the group that is donating the proton. For example, this is our carboxyl group. Let's say the pKa of this carboxyl group is 2 and that of this amine group is 8. So for this carboxyl group to lose a proton, to be in this stage, in this second stage of dissociation, the pKa, the pH of this medium must be greater than 2 for it to dissociate from this stage to this stage. So that is to say, for this to dissociate, the pH of this solution must be greater than 2. So if I have a pH of 2 here, the amino acid will be dissociated by half. What is the definition of pKa of a group? The pKa of a group is the pH at which that group has dissociated by half, by 50%. Let's say so. So if the medium reaches a pH of 2, which is equal to the pKa of the carboxyl group, then at that stage, half of this amino acid will be in this form and half of it will be in this form. That is, this group, the carboxyl group, has dissociated by 50%. That is to say, if I have 100 molecules of that amino acid and I put it in this medium, it reaches a pH of 2, of which my pKa for the carboxyl group is 2. It means 50 molecules of that amino acid will exist with the carboxyl group dissociated. That is, it has lost a proton, it has donated a proton, and 50% will remain the same, that's without being dissociated. So that is what we mean here. Now, going to the second stage, from this stage to this stage, everything still applies. So what has the proton now is at the level of the amine group. Now the pKa of our amine group is 8, and for the amine group to lose its proton to be in this form, the pH of the medium must be greater than 8. So if I have a pH of the medium of 8.5, the amine group will lose a proton and become this. So if I have pH of 8.5, 8.1 and above, I'll have a dissociated amine group. So that is all that is to this concept of pH and pKa. Now, considering that you have this and you are being asked to look for the pI. What is pI? The pI is the pH at which the amino acid has a net charge of 0. That is, the pI is the pH at which the amino acid exists as a deuterion. As we said earlier, a deuterion is a dipolar ion with an overall charge, the net charge of 0. So if I'm asked to look for the pI of an amino acid, after dissociating it, you should take note that the pI is the pH at which the amino acid exists as a deuterion. So let's proceed and see how we can look for pI. Now, pI is the sum of 2 pKa. We don't know yet which pKa they will be, but at the end we will know. So the pI is the sum of 2 pKa's, the average sum, let's say so, of 2 pKa, which is not fixed. So for this first example, we have our amino acid dissociated. First of all, we have to identify our pI, our deuterion, So this is our deuterium here. Now for the pi, that's the deuterium we have identified. We'll have the first pKa here, and we'll have the second pKa here. The first pKa is that of the carboxyl group, the second is that of the amine group. So what will be the pi? It will be the sum of the first pKa, this one here, and the second pKa, that's that of the amine group, divided by 2. So we'll have a pi of 6. So this is the pH at which the deuterium exists. So on to the second example. Here is our pi. As we can see, this is a negative charge here, and here is our positive charge, and summing them will have a net charge of 0. Take note with the progressive nature of the charges. Now let me say regressive. Here you have a +1 charge, here we have a 0 charge, here we have a -1 charge, here +1, here 0, here you have a -1, here you have -2. So if you to know that your dissociation is correct, you should check the charges. If they are decreasing in a progressive manner or in a regressive manner, that's arithmetic progression, you know that it is correct. And it also depends on which medium you are dissociating from. You can either be dissociating from a basic medium, that's high pH to low pH, or from an acidic medium, that's from low pH to high pH. In each case, you should know your net charge should be decreasing in an ordered manner. So here we are in the second stage. In the second dissociation, we have identified our deuterium. Here is the pH for the first carboxyl group that is dissociated, the pKa, sorry. Here is the pKa for the second carboxyl group that is dissociated, and here is the pH for the amine group. So what is our pi now? Our pi is the pH of the first carboxyl group and the pKa, of the first carboxyl group and pKa of the second carboxyl group in that amino acid, the average sum. So 2.7, that is what we have, 2.7. So going to the next, here is our pi in this case with a net charge of 0. Here we have a +2 charge, here we have a +1 charge, here we have a -1 charge. So you see that ordered manner in which the charges are decreasing. So here is the pKa of the carboxyl group dissociating. Here is that for the first amine group, pKa of 9. Here is for the second amine group. So taking the two, calculating the pi, you see that it is pKa of the first amine group dissociating and that of the second amine group dissociating. So that is the pH at which the deuterium of this amino acid exists. So if you look at this trend, what can you say? Because there is a certain confusion that the pi is calculated by pKa1 plus pKa2 divided by 2, which is wrong. What you can observe here is that the pKa's that we're adding and looking for their sum are the pKa's before and after the pi, after the deuterium. So for the first case, you saw we took the pKa before the deuterium plus the pKa after the deuterium to look for the average sum. In the second case, we did the same thing, the pKa before the deuterium and the pKa after the deuterium, and that is what we have. And the third case, the pKa before the deuterium and the pKa after the deuterium. So you see, it changes constantly. It's not the same for every amino acid because the R groups of all amino acids are not the same. There are some R groups that cannot dissociate, while there are others that can dissociate. So that's what makes the difference. So you should take note of that. So we have come to the end of what we had to share today on amino acids, the pKa, the pi, and the acid-base behavior of amino acids. So if you have your questions, please drop them on the comment section below and we'll attend to them instantly. Thank you for watching this video and we hope to have you in our next video. This is Joe Academy and we'll always be there for you. Thank you very much. Please, if you have not subscribed, make sure you subscribe and share our videos. Make sure you like, subscribe, share our videos. If there's any comment, share it in the comment section below. And also we need your reviews, your corrections, your criticisms. We receive all. So thank you and have a nice time.